Hello, everyone. Thank you all for joining our webinar titled N-Nitrosamine Risk Management from Toxicology to Targeted Analysis, presented by Neotron. I'm Elena Caretti, and I will be your host for today's session. If you have any questions in the middle of our presentation, feel free to use the Q&A section or the chat box. We will address those questions toward the end of our webinar. Before we dive into our main agenda, let's introduce our speakers for today. Jonathan Frasca, Manager Director at CMC Pharmatech, who will guide us through the key aspects of risk assessment related to nitrosamines in the pharmaceutical field. Caterina Cappuccini, QC Manager at Neotron, and Sofia Magli, Team Leader at Neotron, will explain the analytical strategy for the detection of nitrosamines. Manuela Pavan, Principal Consultant at Innovatune, will discuss the toxicological evaluation performed after analytical testing to accurately interpret results. For today's agenda, we will talk about an introduction to nitrosamines, and then nitrosamine impurity testing. Finally, toxicological risk assessment of nitrosamines, some case studies, and the Q&A session. Now, a brief introduction about our company, Cotecna. We are leading provider of testing, inspection, and certification services, dedicated to making supply chain safer and more efficient. We are headquartered in Geneva, Switzerland, with offices and lab worldwide. We have over 6,200 employees serving in more than 180 offices and laboratories. Our expertise span across 5 key sectors, namely food and pharma, agriculture, metals and minerals, certification, and ESG solution, and consumer products. Something about Neotron. Here are our key figures of Neotron. We have a turnover which is more than 53 million euros. We have 10,000 square meters of laboratories, more than 550 employees, more than 5,000 accredited tests, and more than 800 methods. And these, subsequently, are our main segments of activity. Historically food, but also baby food, food supplements, food ingredients, cosmetic, R&D, our GMP unit, and food contact materials. Now, I leave the floor to the first speaker of the day, Jonathan, with his introduction to nitrosamines, a risk-based approach. Please, Jonathan, the floor is yours.
Thank you, Elena, for the nice introduction. So I am Jonathan Frasca. I am a chemist by training, and I spent more than 20 years in the pharmaceutical business. Since the last 2 years, we are owner of CMC Pharmatech consulting company, and I was asked to do this first part of the webinar based on the risk-based approach. Next, please. So, starting from nitrosamines, where they came from, many of you for sure remember in 2018, some blood pressure medicines known as sartans contain some nitrosamines. From that point, EMA started a deep review of those impurities, and under Article 5 regulation from the EMA, they provided a guidance for marketing authorization holders to avoid the presence of those impurities. So we know this as call for review. And of course, marketing authorization holders are responsible for ensuring that all products they sell on the market are free of nitrosamines. So next, please. What is the call for review? So we have 3 steps. The first one is the base of my talk. It's a risk evaluation to identify the nitrosamines in APIs, in finished products, as well as excipients. Step 2, in case of positive risk assessment, you have to proceed with the confirmatory testing. and confirm or refute presence of those nitrosamines. And in case you find nitrosamines, then you have to do some mitigation measures to avoid the presence of those nitrosamines. Next slide, please. So structure and formation, how they form. There are many, you can find many articles in the web, but in principle, you have a secondary amine that is treated with the sodium nitrate or any other nitrosating agent. In presence of acidity, you get the nitrosamines. So nitrosation sources are nitrate salts, nitrogen oxides, or nitrocellulose by the degradation. So there are many ways to get nitrates. Also in water, if you use potable water, if you use spring water. So this is really coming from many sources. Next slide, please. So why are toxic? So this is a good question I had in mind at the beginning. So if you look at the metabolism, you have some enzymes that convert the nitrosamine into very potent alkylating or arylating agents. So I imagine some of you has seen a Sandmeyer reaction that is used in the chemistry industry as well as in the pharmaceutical industry to get substituted aryl compounds starting from anilines, but in a similar way, alkyl compounds can be generated during the metabolism in your body. So this is something happening in your body, forming the very potent alkylating or arylating agent. So I have just introduced this as a short introduction since Manuela will talk about this in better details later. But this is the base to say those compounds are quite dangerous. Next slide, please. And about the quantities. So if you take, for example, N-diethyl nitrosamine, we have an acceptable intake of 26.5 nanograms per day. This is a very few. And if you consider why are toxic, so if you consider a body is around 30 trillion of cells, and 26.5 nanograms are around 1.6 14 molecules. This means if you do a brief calculation, you have around 5 molecules of NDEA per body cells. Of course, this is an assumption and this is a worst case, but this can give you an idea why small quantities might be dangerous. If you consider, for example, botulinum has a lethal dose of 100 nanograms, and this calculation can explain why those are so dangerous, those compounds. Please, next. And I would like to give you a consideration about the quantity. So 26.5 nanograms is really nothing. So if you take a crystal of sugar and you divide in 40,000 parts, one part is 25 nanograms, so almost equivalent to the NDEA. And this is really giving me an idea of what about we are talking. So nothing. So really small quantities, and therefore this is what we keep in mind during the risk assessment, because when you do a risk assessment, you have to be really open. So you should apply an holistic approach to think out the box. So next, please. So possible contamination during your process. Secondary and tertiary amines, of course, you can have those in starting materials, used as solvents or reagents. It could be impurities in the same solvents. If you use, for example, triethylamine as a reagent or as a solvent, you could also have diethylamine, etc. So these are coming from really many sources. Nitrosating agents, as we saw before, nitrates, nitrates used in the process, coming from water or excipients or APIs as well. Acidity, you can find really many parts. So acids added to the process, excipients like citric acid, API itself as hydrochlorides or other acids. So these are just few examples, but you should really look into every single component to understand if you have one component present. Next, please. So as I said, a holistic approach. You should start thinking about the process life cycle of your compound. So starting from the route of synthesis for starting materials, raw materials, reagents, solvents, giving a look, seeing what is used in the process or in early processes, specification of raw materials, because, for example, you get a product from your supplier, which is in spec, but then are the spec adequate for their intended? So I remember one case where we had triethylamine as potential solvent or reagent inside an excipient, and these had a specification of 100 ppm. By doing some calculation, this was not enough to guarantee free of nitrosamine in the final product. And of course, you have to think about all the life cycle of the process, so the API as well, the finished product. So you should consider where starting materials are entering the process, if secondary tertiary amine are entering, if in the process are places where nitrosating agents are and acidity is combined with every single components to have the condition to get the nitrosamine. So all process steps should be considered. I would think about phase cuts or when you mix different powders in the finished product. So you have really many opportunity to have this combination. And it's not just a liquid phase problem, it is an overall problem because you can have also in solid state. So you should also consider when you mix APIs and excipient together, if you have the combination of all components to get this multiple factor, multiple contribution and get the nitrosamine. Packaging material also, if you use packaging material with nitrocellulose and you have a thermal closure, then you could have degradation of the plastic and get some nitrates from the packaging material. So this is really everything. You should really consider every single step. So next, please. So starting from a risk assessment, you may use different tools. You can use a failure mode effect analysis. You can use fishbone diagram, decision tree. So I mean, any tool is valid. It's up to you to use a good tool, and it should be really used systematic on every step, every component, and the mixed solutions, mixed powders to understand if you have some issues during your process. Of course, it is an interdisciplinary team, and you should involve trained people for the assessment, with also in mind that we are looking for very small quantities with high potential in toxicity. Next, please. This is an example of decision tree I prepared. This is usually what I use in my mind, but it's just an example. It's not, I would say, not a rock, not written the rock, but you can adapt your tree as you want. But in principle, you should consider all components. So, for example, here we have you take your components that you have checked with your supplier, knowing that they are free of nitrosamine, using questionnaires or other tools. Then you start your review, say, okay, we have secondary, tertiary amine. If no other sources from packaging material or not, then if no, you can say no further action. In case you have the amines present, then you can consider nitrate are present. If no, again, packaging material, and if no, you can conclude you don't have issues. Last point, of course, once you have tertiary amines, secondary amines, and nitrate present, you should consider if you have also acid conditions, because this is another point you need that in case you have all components, of course, you should then understand the nitrosamine's presence, and then you have to start with the step 2. So development of an analytical method and check the presence of those impurities. Next, please. I am concluding my part by sharing with you a case study I had during my previous previous work. So we had a finished product with multiple APIs, and looking at the questionnaires from the vendors, all were free of nitrosamines. Then same thing for excipients, all were free of nitrosamines. And based on this, the customer said the initial evaluation is okay. We can say there are no nitrosamine in finished product. However, they did not consider the contribution of the single component, single API, and the excipients together, and the authority asked to a better evaluation, a deeper evaluation. And therefore, they came to me asking for this. Next, please. What I found during my review, one API contained a tertiary amine as a reagent in the synthesis, and this was a first, let's say, point. Second point was another API contained a secondary amine as functional group, an impurity, and this was a second point of alert. Going ahead with this evaluation, I found nitrates in an excipients that they use pre-generatized starch. So they had the nitrates present in the process. And the acidity was granted by other components. One was an API as hydrochloride salt, and another acidity came from an excipient. So in the end, we found out that two potential nitrosamine could be formed. And therefore, the recommendation was to check with the specific analytical methods, because of course, the methods in place were not able to detect them. Next, please. So concluding this first part of the risk assessment, this is really important to be done in a certain way. So you should consider really all single components used in your production, the combined components, to see if amines, acidity, and nitrates or a nitrosating agent could be present at the same place in the same process. All process steps to see, for example, mixing of this solution or mixing of different solids could bring you in a bad position. And of course, using multi-purpose plants should be verified for crosses of different products. In this case, I have an example in my previous slide that we had one product using dimethyl as a solvent for the cleaning because it was really insoluble, and therefore dimethyl had to be evaluated for the next product because it was quite acid, and under acidity you could form dimethylamine. So this is what I recommend you to do in such studies. And if you have any questions, I'm here. I give back the word to Elena. Thank you.
Thank you, Jonathan, for your contribution. It was really interesting and much appreciated. Now I leave the floor to Caterina Cappuccini and Sofia Mali from EMA updates on nitroso APIs to real case studies. Please, Caterina, the floor is yours.
Thank you, Jonathan, for your presentation on the risk assessment. Now I will explain how to approach with the analysis of the nitrosamine. I am Caterina. I work with Sofia in Neotron Pharma, that is the pharma division of Neotron, that is part of Cotecna, the corporate, as Elena explained before. Next. Neotron Pharma is authorized by AIFA, the Italian Medicine Agency, the Italian Ministry of Health for the usage and detention of psychotropic substances, and by FDA. Next. We cover several analytical activities as chemical and physical test according to Pharmacopoeia, European, USP, Japanese, test for impurities and contaminants, test according to registration. We develop and validate analytical method with several analytical techniques, and we offer stability storage services and stability studies. Next. But in what our laboratory can support for the analysis of the nitrosamine? So we can support with the risk assessment, the method development for API and drug product specific, stability study, drug product quality and shelf life, evaluation of the interaction between API and excipients, and what is the general approach. So the customer comes to us with a request, we check the availability of the standard, we develop the method, and then apply this method for screening limit test or full method validation. Next. The availability of the standard plays a very big role. We collaborate with a specialized company that have in stock the standard, or they have to synthesize the standard for us. Unfortunately, sometimes can take also 2 or 3 months to receive the standard. So once we have the standard, we can start with the development of the method. Next. It's very important that the supplier of the standard gives all the information about the product, characteristics, solubility, the mass spectra to compare to the one that we obtain during mass optimization, and the indication about chromatography, because it's a very good starting point for the setup of the method. Next. When the standards cannot be synthesized, maybe we can evaluate other paths, and other paths are available. For example, in this case, the customer asked us to investigate the possibility of having nitrosodisulfiram in their product. The supplier could not isolate the nitroso derivative API because the API was not stable under acidic conditions and transformed directly in the correspondent thiols, in this case nitrosodiethylamine. So from the collaboration between the customer, the supplier, what we have validated in the end was a method for the detection for the nitrosodiethylamine. Next. We have the standard, a suitable technique has to be chosen, usually LCMS, because it's much more sensitive compared to other techniques as GCMS or TOF. We go through the optimization of the mass parameter and chromatography, so the molecular ion characterization. We try different chromatographic column, different mobile phases. We set the perfect gradient in order to separate the API with the nitrose API, and we optimize mass parameter. A very critical step is the sample preparation, because depends on the pharmaceutical form, and it's important to find the correct solvent of dilution for the API. So it's suggested when we are in front to develop a method to work separately on the API, on the placebo, and the finished drug product in order to optimize the method perfectly. Next. The document question and answer of EMA gives indication on several requirements. For example, the question and answer number 9 explains the requirements of the analytical methods. An analytical method has to be sensitive, accurate, and repeatable, and gives a definition of the limit of quantitation that provides the level at which an analyte can be quantified. In the case of routine methods, the limit of quantitation has to be below the acceptable intake. In the case of skip testing, between the 10% and the 30%. In the case of omission of specification, below the 10%. Next. Appendix number 1, always of EMA documentation, contains all the nitrose API, so large molecules, and the nitrosamine small molecules that has to be controlled. Now they are more than 200. The acceptable intake of each nitrosamine is a fundamental information to calculate the limit of specification. The limit of specification is calculated dividing the acceptable intake of the nitrosamine for the maximum daily dose of the API, and the 10% of the limit of quantitation we were talking before is the 10% of the limit of specification calculated as just explained. Next. At this point, when the method has been developed, we have two paths, quantitative method or qualitative method. Usually it's suggested to start with a qualitative method that can give a clue, a general info on the entity of contamination that we have in our product. The limit of detection is preferable to be set at the 10% of the limit of specification, but unfortunately sometimes the limits are so low that is very challenging for the instrument sensitivity. But the documentation collected, if the contamination is below the 10%, can be sometimes enough for the authorities. If the contamination is present, has to be quantified, so we have to proceed with a full validation method according to the ICH Q2R2, so all the tests, linearity, accuracy, repeatability. Now I leave the stage to Sofia that will present a real case studies.
One particularly interesting case study involved a request from one of our customers who required a fully validated analytical and quantitative method specifically tailored to a specific commercial product. The goal was to establish a standardized and reliable approach for identifying and quantifying the analyte in several critical applications. First of all, the analysis of different product batches, including those exhibiting high levels of nitrous API contamination. The evaluation of binary mixture formulations to investigate potential interactions between the nitrous API and the excipients present in the final drug product, and finally the support of stability studies performed on the finished drug product throughout its shelf life. Next slide, please. Specifically, the request concerned the development of an analytical method for nortriptyline hydrochloride capsules. As many of you know, nortriptyline API contains a secondary amine functional group which can react with nitrosating agents, typically nitrites, to form N-Nitrosamine, the corresponding nitrosamine impurity. During the initial screening limit test, the finished drug product showed, as expected, a high natural content of the analyte far above the established specification limit for nitrosamine. However, when the intrinsic level of an analyte in the drug product is already very high, it becomes challenging to validate the method and to properly evaluate performance at the limit of quantitation level. For this reason, our proposal was to perform the full method validation on a placebo matrix formulated exactly like the final drug product but without the active ingredient. The method was therefore validated across a wide linearity and accuracy range capable of accommodating high analyte concentrations. Once validated, the method was subsequently verified on both the finished drug product and the nortriptyline API itself. This verification phase included accuracy and repeatability assessments at both the limit of quantitation and the limit of specification to ensure robustness of the method across the entire analytical range. To identify and reduce the potential sources of contamination, the validated method was subsequently applied to study API excipients interactions using binary mixture formulations provided by the customer. We first assessed whether there were any differences in nitrosamine contamination originating from two different nortriptyline API suppliers, referred to here as API A and API B as shown in the table in the slide. For both suppliers, the nitrosonortriptyline content in the API as is was found to be below the limit of quantitation, indicating that the API itself was not the primary driver of the contamination. As a second step, we investigated how the two API types, A and B, behaved when combined with the two key excipients present in large quantities in the final capsule formulation, anhydrous lactose and the pre-gelatinized starch. These binary mixtures were evaluated under both non-stressed and thermally stressed conditions to capture potential formation trends under realistic and accelerated scenarios. In parallel, we complemented the nitrosamine study with a nitrite determination in the excipients using ion chromatography technique. The results clearly showed that anhydrous lactose contamination, making it the most critical excipient in terms of promoting nitrosamine formation for both API batches. In contrast, pre-gelatinized starch contained only traces levels of nitrites from its manufacturing process, contributed far less to nitrosamine formation. Just to briefly sum up, different excipients contribute differently to the formation of N-Nitrosamine drug substance-related impurities. Excipients containing nitrites, even in traces, are considered high-risk excipients due to their ability to react with the secondary amines. API synthesis routes from different suppliers can influence nitrosamine formation. Although both API A and API B were below the limit of quantitation in their raw state, differences in synthesis or impurity profile may still affect their behavior in formulation. This information is valuable for customers when selecting the most suitable API supplier. Finally, stress conditions accelerate nitrosamine formation, confirming that environmental and processing conditions play a significant role in the overall risk. In this regard, we conducted the stability analysis on the final product formulation, monitoring the content of N-nitrosonortriptyline over a 24-month stability period. Stability studies performed in climate chambers where temperature and humidity are continuously controlled allow us to observe whether nitrosamines form during storage and whether their levels increase over time. These studies may be carried out directly by the customer, outsourcing only the impurity determination to us using the validated method, or they can be fully managed by our laboratory, including both sample storage and analytical testing. Stability studies over 24 or 36 months allow the standard approach defined by guidelines, which require long-term experiments to determine product stability under realistic storage conditions. These studies are specifically designed to establish the shelf life of the drug product and to ensure that the finished dosage form maintains its quality, safety, and efficacy throughout its intended storage period. As shown in the results tables, the formation of the nitrosamine drug substance-related impurity persists throughout the product's shelf life. This behavior is expected given that the API contains a secondary amine and the formulation includes excipients with measurable nitrite content. Under 25 degrees and 60% humidity conditions, we observed a gradual but modest increase in nitrosamine levels over time. Under 30 degrees and 75% humidity conditions, the increase remained modest but clearly detectable. And finally, under 40 degrees and 75% humidity conditions, the increase was significant and evident, confirming that the strong influence of temperature and humidity on nitrosamine formation. Next slide, please. In conclusion, let me summarize our key take-home messages. Modern pharmaceutical regulation is evolving. The regulatory landscape has progressively moved away from statics, compliance-only oversight toward linear, science-driven, and efficacy-oriented frameworks. This evolution actively encourages and in many cases requires continuous process improvement throughout the entire pharmaceutical life cycle. Process improvement requires strong analytical support. Analytical support is therefore a critical component of pharmaceutical safety. Validated analytical methods play a key role in standardizing manufacturing processes and enabling the reliable evolution of all batches from raw materials to final dosage forms. They also support stability studies, supplier qualification, impurity risk assessment, and decision-making beyond the initial formulation stage. Together, these elements demonstrate that analytical science is not merely a testing step, but it is a strategic enabler of product quality, regulatory compliance, and patient safety. Next slide, please. And thanks for your attention and your time.
Thank you, Caterina and Sofia, for your contribution. Clear, to the point, and really useful for everyone. Now I leave the floor to Manuela Pavan, Toxicological Risk Assessment of Nitrosamines, Navigating Scientific and Regulatory Challenges. Please, Manuela, the floor is yours.
Thank you, Elena, and thank you to all of the participants for the interest in the webinar. Today, I will discuss the toxicological risk assessment of nitrosamines with a focus on the underlying scientific principles and the regulatory frameworks that are applied, particularly in challenging cases where substance-specific data are unavailable. Nitrosamines are a structurally diverse class of compounds characterized by the presence of a nitroso group attached to the nitrogen atom of an amine. They are, as already anticipated and presented in the webinar, they are typically formed through the nitrosation of secondary or tertiary amines, and nitrosamines are well known to be mutagenic and potential animal carcinogens. Within the ICHM7, which provides the framework for the assessment and control of mutagenic impurities in pharmaceuticals, nitrosamines are included in the so-called court of concern, a group of highly potent carcinogens for which default threshold-based approaches do not apply. This court of concern also includes other highly potent classes of nitroso compounds, reflecting their shared concern from a toxicological perspective. Next, please. So, to understand their risk, it's essential to understand their mechanism of action. Nitrosamines are not directly DNA reactive. Instead, they require metabolic activation primarily mediated by the cytochrome P450 enzymes. And the key step is an alpha-hydroxylation, which leads to the formation of an unstable intermediate that rearranged into a highly reactive diazonium ion, which is capable of alkylating DNA. This DNA alkylation may be repaired by cells' DNA repair systems. However, if it is left unrepaired, it can be propagated during the DNA replication, leading to mutations. And over time, the accumulation of such mutations may contribute to cancer development. So this slide illustrates the underlying chemical reactions that are involved in this mechanism. As we said, the bioactivation is an indirect process that requires metabolic conversion by liver enzymes and potentially enzymes from other tissues to generate the ultimate carcinogenic species. So the critical step is really this cytochrome P450 catalyzed alpha-hydroxylation, which initiates a series of reactions that are leading to the formation of a highly reactive diazonium ion able to alkylate DNA. And this mechanistic understanding explains why nitrosamines are potent carcinogens across a wide range of animal species, including humans, as the cytochrome P450 enzymes involved are present across the species. Secondly, this mechanism also explains why structure differences among nitrosamines are so important and matter. Structural features that make the alpha hydroxylation less likely or that favor alternative detoxification pathways generally lead to lower carcinogenic potency. So this mechanistic understanding provides really the scientific basis for many of the regulatory approaches that are currently used. Next, please. Building on this mechanistic insight, the literature has identified several features, structural features that can modulate the carcinogenic potential of the nitrosamines. And these structural activity relationships or features include pharmacokinetic factors. For example, carboxylic acid groups may promote faster clearance from the body, reducing the systemic exposure. Steric hindrance, so if we have bulky groups near to the alpha carbon, this can physically block the cytochrome P450 enzymes, thus limiting the bioactivation step. Or electronic effects. Electronic withdrawing substituents can lower the likelihood of a diazonium anion formation and therefore decreasing the potential for the DNA alkylation. So these features help explaining why some nitrosamines are more potent than others and are key considerations in the scientific evaluation of their risk. Because of their mechanism of action, nitrosamines exhibit a high carcinogenic potency and are included, as we mentioned, in the court of concern under the ICHM-7 guideline. And as a result, they cannot be managed using the default TTC value of 1.5 microgram per day applied to most mutagenic impurities, but instead they require compound-specific acceptable intake limits. When sufficient substance carcinogenicity data are available, the M7 recommends deriving these limits based on the TD50 values, where the TD50 is the chronic daily dose associated with tumor formation in 50% of test animals. And for some well-known and well-studied nitrosamines, such as NDMA, NDA, extensive rodent carcinogenicity data are available, and these allow acceptable intake values to be derived directly from the TD50 data. And reliable TD50 values can be obtained from databases such as the carcinogenic potency database or proprietary resources like the LASA carcinogenicity database. However, many nitrosamines are data poor, meaning that, and specifically this is the case for those that are derived from the neutralization of amines in drugs, so the so-called nitrosamine drug substance-related impurities. And this creates a key challenge for risk assessment. How can we derive the acceptable intake limits when substance-specific data are limited or not available? In fact, from a toxicological perspective, nitrosamines can be broadly divided into two main groups. The first includes small alkyl nitrosamines like NDMA, NDA, and these compounds are generally well studied and supported by extensive rodent carcinogenicity data. The second group consists of the nitrosamine drug substance-related impurities that are generally more structurally complex, and that often lack robust toxicological data, and for this reason they are commonly referred to as data poor nitrosamines, and these are those posing particularly challenges for risk assessment. So to address these gaps, 4 alternatives, science-based approaches have been established by EMA and also other regulatory authorities. One widely adopted method is the CPCA, the carcinogenic potency categorization approach, which groups nitrosamines into potency categories with associated acceptable intake limits based on structure features and structural alerts. Another option is the use of a negative result from an CLP-compliant enhanced Ames test. Another approach is the read-across. So again, if a structural surrogate nitrosamine has well-characterized carcinogenicity data, this can serve as a point of departure for deriving the acceptable intake limit of the data poor nitrosamine. And finally, if nitrosamine gives a negative result in a well-documented in vivo mutagenicity study, this can be treated as not mutagenic impurity, and in that case, it can be controlled under ICH Q3A and B, regardless any AI calculated using a CPCA or read-across. While EMA and FDA recommended broadly similar strategies, there are also some differences. And I noted that there was a question, I think, in the chat, and I think that hopefully this slide is also answering that question. So there are some differences. With respect to the CPCA classification, the approach is almost the same. The only difference is related to the lowest limit that is set at 80 nanogram per day by EMA, while it is 26.5 by FDA. With respect to the enhanced AIMS test, both agencies accept a negative result to support a higher limit, but FDA typically requires additional data. Read-across is almost accepted in the same way, while with respect to, again, in vivo mutagenicity studies, while EMA accepts a negative result from a well-conducted in vivo study to justify and control the impurity as a non-mutagenic impurity, the FDA does not consider such data alone sufficient to support a qualification threshold defined as in Q3A and B. So the CPCA approach, as we mentioned, is a prediction of the carcinogenic potency of a nitrosamine that can be generated taking into account a number of different elements. We consider in this approach the number of alpha hydrogen, which determines how readily the compound can undergo alpha hydroxylation. It's also based on some activating features and also deactivating features. And all these three components are assigned to a score and then sum up to derive the total potency score that is then used to assign nitrosamine to a given potency category. So this slide shows the flowchart that is used in the CPCA decision tree. There are a number of structured questions with the calculation of the potency score that is then used, as I said, to derive and to assign the nitrosamine to a given category. This flowchart can be applied manually, but is also implemented in a number of software tools. And by following the decision tree, it's possible to define the potency score of a nitrosamine of interest and derive the corresponding acceptable intake limit. So here is just it's just an example. So we considered here the same example that was discussed before. So the case of a nitrosonotriptyline. The first step is always to verify whether an acceptable intake limit has already been established by regulatory authorities. And in this case, we have a limit that has already been reported by EMA, but also by other regulatory authorities. It's important also to note that the limits reported, for example, in the Appendix 1 of EMA may be derived by different approaches that can be based on a CPCA, read-across, or in vivo mutagenicity data. For this specific example, the acceptable intake is derived based on the CPCA approach. And here you can see how it was computed, so how the potency score was computed, and how also the decision tree applies to that specific nitrosamine that at the end leads to the assignment to the highest carcinogenicity potency. In the case where a regulatory acceptable intake values are unfeasibly low, applicants can still propose alternative values supported by robust scientific justification. And this includes results from enhanced AIMS test, but also read-across and in vivo genotoxicity data. Next, please. Read-across is an approach that is widely used to fill in data gaps in human risk assessment, and it is based on the concept that it's possible to estimate the endpoint of a target substance using data from the same endpoint from another substance, which is data-rich, based on a robust justification on the mechanistic underlying leading to the toxicity. In the context of nitrosamines, the read-across is a case-by-case approach that is used to derive acceptable intake limits, and the approach, as I said, is supported by similarities assessment in structure, but also physical-chemical properties, ADME, and special metabolism, and also chemical and biological reactivity. And this slide outlines the typical workflow that is applied for the read-across. So we firstly need to identify suitable analogs, then the quality of available carcinogenicity analog of is evaluated, and the assessment of similarity is also key in the approach. And at the end, if an analog meets the criteria across these domains, then a reliable TD50 value can be used as the point of departure to derive the acceptable intake limit of a nitrosamine. So health authorities, this is just very quickly to say that health authorities have applied this approach to derive substances acceptable intake limits for data poor nitrosamine. And here are just some examples that I leave it to you. So to conclude, nitrosamines are a class of highly potent carcinogenic impurities that require a case-specific and science-based risk assessment. The mechanistic understanding and the mode of action is essential and provides the foundation for predictive approaches such as CPCA and read-across. And these approaches allow the derivation of scientifically justified and defensible acceptable intake limits, even in situations where substance-specific data are limited. And ultimately, the risk management is, in this case, is not about achieving a zero risk, but is about defining and controlling an acceptable level of risk. And with this, next, I would like just to thank you for your attention and also to thank all my colleagues for their support.
Thank you, Manuela, for your presentation. Clear and really helpful. Following our speaker's presentation, we'll now open the floor for the Q&A session. We received these questions during the registration process. So the first one, please. Is there any regulatory or scientific preference among CPCA, read-across, and the enhanced aim test for deriving acceptable intakes for nitrosamines? I think, Manuela, this can be for you.
Yes, thank you. Well, I have to say that there is not a universal scientific preference because the most appropriate approach really depends on the data that are available for a given nitrosamine. CPCA is generally the starting point, then read-across may also be valuable only in the case we have a good analog with a good carcinogenicity data. And of course, the AIMS test has a different role if you have a negative outcome that can support, for example, a TTC level. But if it's possible, you still need to go through CPCA or read-across to derive a limit.
Thank you, Manuela, for your clear answer. Now, question number 2. What is the most important thing of a company should consider in the risk assessment? Jonathan, this is for you.
Thank you, Elena. I think first thing you need a tool, a systematic tool to do the risk assessment, to check all components, all parts of your process. But I think most important is to have the right people. So people make the difference because you need trained people to do that, good chemists to see the process and to think out of the box. Because if you concentrate on the main process, you'll never find those nitrosamines. You have to think a little, and in this case, details make the difference.
Thank you, Jonathan. Question number 3. Why can different nitrosamines show such large differences in carcinogenetic potency? Manuela, this is again for you.
Thank you. Well, the main reason is that the nitrosamines require the metabolic activation to become DNA reactive, and the carcinogenicity potency really depends largely on how easily they can undergo the alpha hydroxylation and form the reactive diazoxime ion. So the potency is not determined only by the presence of the group, but rather on how efficiently the molecule can be metabolically activated to a DNA reactive species.
Thank you, Manuela. Now, question number 4. What are the main pathways for nitrosamine formation in APIs, finished products, and drug during storage? Sophia, this question is for you.
Yes, thank you. Nitrosamine formation essentially comes down to one simple concept. Whenever nitrite meets a nitrosable amine, usually a secondary amine, there's a possibility for nitrosation. What makes the topic challenging is that this interaction can happen at several points in the life cycle of the drug product. During API synthesis, nitrosamines can form because trace amounts of nitrite may be present in raw materials, reagent, or even process water. If these traces encounter an amine, or if nitrite converts into nitrous acid under mildy acidic conditions, nitrosation can occur even at very low levels. This means that even small impurities or slight pH variations can be enough to trigger the reaction. In finished drug products, the main contributors are the excipients, many of which naturally contain nitrites like microcrystalline cellulose, magnesium stearate, talc, starches, or povidone. These nitrites can react with APIs containing amine groups. Nitrites can also be converted into nitrites in the presence of metals or reducing agents, and in liquid formulation, acidic microenvironmental can significantly accelerate the formation of nitrous acid. In certain cases, even packaging materials such as rubber closures may release nitrosating species that migrate into the product. And finally, during storage, nitrosamines can still form over time. The API might slowly degrade into amines, excipients may generate new nitrite, and oxidative conditions like exposure to oxygen, humidity, or light can create reactive intermediates. It also plays a major role because elevated temperatures speed up both the degradation pathways and the nitrosation reaction itself.
Thank you, Sophia, for your clear explanation. We received many other questions. We cannot reply to all of them. I consolidate all these questions and they will be covered with a follow-up mail. As we near the conclusion of our program, we'll send out an email in coming days containing a survey form and a recording of this webinar. Additionally, for more news and updates about Cotecna, we will be providing a link to our newsletter subscription. You may also reach out us using the contact information provided on your screens. We hope you find this webinar beneficial. A big thank to our speakers for sharing their expertise and to all of you joining us. Have a great day, everyone, and see you at the next webinar.